Okay, another brief introduction to the topic. So if you attended our Science Olympiad class, you know a bit more about genetics. Just know that the father of genetics is Gregor Mendel, and he experimented with lots of things. He came up with these ideas of genes and alleles, and genes are the unit of heredity, and they're passed on from generation to generation. They determine all sorts of different traits, like, for example, your hair color or your eye color. And these genes are carried in DNA, and this DNA associates to become different chromosomes. And in humans, we have 23 pairs of chromosomes, 22 somatic chromosomes, and then one sex chromosome pair. And then just know these terms like genotype and phenotype, right? The genotype is which alleles that you have. The phenotype essentially is based off of your genotype. What are your actual traits or appearances? Punnett squares, which most people will learn in biology in middle school, is like how you determine what the genotype of a descendant will be. There are two different types of traits and diseases, like autosomal versus sex-linked, right? And then just remember that sex-linked means that your sex, whether it's like male or female, determines what expression the trait is. And common examples are hemophilia and color blindness, and those are normally carried on the X chromosome. So we say sex-linked. Most of the time we mean genes that are on the X chromosome, just because the Y chromosome is so small and it's much less likely to carry any traits. And then there are two types of alleles that Mendel hypothesized. He talked about dominant alleles and recessive alleles. Dominant alleles are alleles that are kind of just like, when you have a dominant allele, you express the dominant trait. And when you have a recessive allele, you have to have two recessive alleles to express the trait. So if you have a dominant allele and a recessive allele, you'll express the dominant trait. It dominates over the recessive trait. But then Mendel also didn't know about different forms of non-Mendelian inheritance, which are the forms of inheritance that he did not speculate on. And so two forms of that: codominance, which is when you have alleles that are both dominant and they just show up together. So that would be like the ABO blood system, right? If you have the A allele and the B allele, they don't dominate over each other. You just get the AB blood type. You get both. And incomplete dominance, where the dominant allele is not completely dominant over the recessive. So if the dominant allele is like red flower, and the recessive allele is gives a white color to the flower, and then you have both the dominant allele and the recessive allele. Normally, Mendel would expect, right, the red is dominant, white is recessive. So a heterozygote, which means it has two different types of alleles, the heterozygote would just display the dominant characteristic of being a red flower. But that's not the case for incomplete dominance. It's going to be like a halfway kind of phenotype. So it's going to be a pink flower instead, because the dominant allele is not completely dominant over the rest of the alleles. Okay, and so now let's take a deep dive into cell reproduction. And so this form of cell reproduction is called mitosis. Remember, it's called mitosis. You can see a very, very similar name, but it's gonna be very different in the future. So mitosis has to do with if you have one cell and you want to get two. So you have this mitosis in a bunch of different phases. The first phase, which is not really part of mitosis, is interphase. This is just when the cell is not in mitosis. And in interphase, the cell is like growing, it's doing all sorts of different things that the cell would normally do. And then the chromosomes also in the middle of interphase, they're going to be duplicating, so preparing for reproduction, right? If you just have your chromosomes and you keep dividing them in two every time you reproduce, pretty soon, right, you can have the original cells have the normal amount of DNA, and the next generation will have half of the DNA, and then the generation after that will have 1/4 of the DNA, and then your cells would die. They have all sorts of weird mutations, they wouldn't survive. So the first thing you need to do is, if you're going to split your cell in half, you need to double your DNA content first, so that each of your daughter cells, which we call the products of mitosis, each of your daughter cells will have the same amount of DNA that you originally did. So your chromosomes duplicate in interphase. And then when you enter mitosis, you're finally preparing to essentially actually split your cell into two. So your chromosomes, they become very condensed and visible. This is what you normally see when you think of chromosomes. You think of like these like thick X's. Normally chromosomes are actually just like a blur, a blob of like, looks like string. But in prophase, they're going to condense together and form these very recognizable chromosomal structures. And then remember that DNA is contained in the nucleus, right? But if you want to split your cell in two, you're going to have to kind of get rid of this nucleus. So this nucleus dissolves in prometaphase, which some books don't consider its own unique phase. Some books say prometaphase is part of prophase. But for now, we're just going to consider prometaphase as something unique. And so in prometaphase, the nuclear membrane, the nucleus breaks down. So now all these chromosomes are going to start just drifting in the middle of the cell. And then the next step after prophase is metaphase. So in metaphase, the cells are going to line up in the middle of the cell because they're preparing for division. So the cells used to just be inside this nucleus, and then now the nucleus broke down in prometaphase. The cells are just floating around. and they're going to align themselves in the middle of the cell in metaphase. And then once they've finished aligning, they get into anaphase, which is the quickest part of the cell cycle. And so in anaphase, the cells, sorry, not the cells, the chromosomes are going to be split in two. So remember, we doubled our chromosomes here, and now we're going to have them. So in the end, we have, this is the original amount of DNA the cell has, and we're going to have kind of two sets of it in telophase. So now you have like two different nuclei in telophase. The chromosomes are separated in anaphase, and now you have two sets in telophase, and these nuclei are reforming around them. And then the last step is just cytokinesis. You're going to split the cell in two, and ta-da, you have two new cells that should be genetically identical to the original cell now. So again, you duplicated the DNA essentially. You have all the DNA lined up in the middle, and then you split the chromosomes, and then they each form their own new nuclei, and the cell splits in half in cytokinesis, and it makes two new cells that have the same genetic identity. So we just finished talking about mitosis, and now we're going to talk about meiosis. So these are very close sounding words, but they're very different in a sense, and very similar in another sense. So we're going to be talking about what are the differences between them, because you'll probably need to remember the unique features of each one. So again, review mitosis. We said that there are these phases. The main ones you want to know: prophase, metaphase, anaphase, telophase, in this order, PMAT. And what you're doing is you're taking one cell and you're making two identical daughter cells. In meiosis, the goal is slightly different. You have this one cell, and you want to make these 4 unique individual sex cells now. So why would we need to do that? So the main difference in meiosis is that instead of making an identical copy, so here we doubled the DNA, and then we're just trying to make 2 versions of the same original cell. But now we want to make sex cells. And so if you just like stop for a second and hypothesize how would reproduction work, right? If you just took 2 normal cells and you stuck them together, then the daughter cell would have twice as many DNA. And as we talked about in the answer clarifications, Q&A clarifications, if you had twice as much DNA, you would not be alive if you're a human. Now, in response to someone else's question about just in general, if you had twice as much DNA, a lot of plants can survive with twice as much DNA. That's how a lot of speciation occurs, but not animals. So we're focusing on animals right now. So in meiosis, we're going to have these cells, and there are 2 phases of meiosis. We have meiosis 1 and meiosis 2. So it's taking step in 2 phases. And the orders are exactly the same: prophase, metaphase, anaphase, telophase. This is kind of more out of order because this is from a worksheet. But the first step in prophase, metaphase, anaphase, telophase, prophase 1, metaphase 1, anaphase 1, telophase 1. What the cell is doing is it's pulling these chromosomes apart, and it's going to actually have the DNA content. So originally, remember we said we had 23 chromosome pairs. And then in meiosis 1, we want to reduce that. So there's just 23 chromosomes. We're getting rid of the pairing. Normally, the pairing is coming from the fact that you have one father and one mother. So you have 2 of each of our 23 chromosomes. But now you're preparing to be a parent. You're making sex cells. So you get rid of one. You're separating the pairs up into 23 chromosomes each. And then you just go through meiosis 2, which is the exact same thing as mitosis. And what you end up with is a cell, instead of having 23 pairs of chromosomes, it just has 23 chromosomes. And so now when you have fertilization, when you combine an egg with a sperm, 23 plus 23 gives you 23 pairs, and you'll have a normal offspring.